Hello everyone, welcome to Ibdp Biology. Today our topic is going to be about membrane transport. So let's get started. Cell must maintain homeostasis in order to live. What is a homeostasis? A process by which a constant internal environment is maintained despite changes in the external environment. So in other way, it means that whatever happens in the external environment of the cell, homeostasis keep the interior constituent of the cell stable. Cells need to bring in food, water, oxygen, and eliminate waste to maintain homeostasis. To do this, cells need to transport of molecules in and out across the plasma membrane, which acts as a barrier. The phospholipid molecules which make up the plasma membrane are in a fluid state, which allows them to change shape. You know from the previous lesson that there are the hydrophilic phosphate heads and made of the hydrophobic hydrocarbon tails. This fluidity of the plasma membrane allows it to form vesicles that can be pinched off or ones that can fuse with. And during endocytosis and exocytosis, these vesicles are formed without any damage to the plasma membrane. So let's get to know what are the endocytosis and exocytosis. First, we will start with the endocytosis. Endocytosis occurs when a plasma membrane is pulled inward, such as this, as you see. The plasma membrane is actually pulled inward and creates a pocket around the particular substance. So it can be a food, it can be about water. So these kind of things, when they enter to the cell, so the plasma membranes are actually pulled inward and create vesicles. So the cells can bring in solids and liquid using this process. So vesicles can taken in by endocytosis contain water and solids from outside the cell, but they also contain larger molecules needed by the cell that cannot pass across the plasma membrane. For example, in the placenta, proteins from the mother's blood, including antibodies, are absorbed into the fetus by endocytosis. And some cells take in large undigested food particles by endocytosis, such as a food vacuole, as you see here. So there are different types of the endocytosis, so let's see them. Pinocytosis, a type of endocytosis in which the cell ingests extracellular fluid and its dissolved substances. And phagocytosis, and phagocytosis actually happens in the white blood cells that they take in pathogens including bacteria and viruses by endocytosis, by phagocytosis, and kill them as a part of the body response to infection. Also, we can see phagocytosis process in unicellular organisms including amoeba and paramecium. It's a type of endocytosis involving large particular substances accomplished mainly by macrophages, neutrophils, and dendritic cells, as you see. Endocytosis, the cellular uptake of macromolecules and particulate substances by localized regions of the plasma membrane that surround the substances and pinch off to form an intracellular vesicle. So remember that endocytosis is actually the plasma membrane which is pulled inward and creates the vesicles inside. Exocytosis. Exocytosis, internal vesicle will fuse with the plasma membrane and the contents of the vesicle are released in the external environment of the cells. The cells can secrete substances they produce this way or excrete waste products, such as we see in the Golgi apparatus. In the Golgi apparatus, when the and rough endoplasmatic reticulum send the proteins to Golgi apparatus to let's say to work on this one, then the Golgi apparatus also create the lysosome and certain proteins. So these are going out from the Golgi apparatus and creates the vesicles, which are we call the exocytosis, because these vesicles are going out of the cell to different cell types where they need a certain substances. Digestive enzymes are released from gland cells by exocytosis. The polypeptides in the enzymes are synthesized by the rough endoplasmic reticulum, processed in the Golgi apparatus, and then carried to the membrane in vesicles for exocytosis. In this case, the release is referred to as the secretion because useful substance is being released, not waste product. Exocytosis can also be used to expel waste products or unwanted materials. An example is the removal of excess water from the cell of unicellular organism. The water is loaded into a vesicle, sometimes called a contractile vacuole, which is then moved to the plasma membrane for expulsion by exocytosis, and this can be seen quite easily in paramecium using a microscope. So at the exocytosis, the cellular secretion of macromolecules by the fusion of vesicles with the plasma membrane. Now I'm going to just show you the video how the endocytosis and exocytosis actually happen. So let's see.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use three basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
Okay, so there are also questions that you can do and submit answers to see whether you're right or not, or we're gonna do it during the lesson time. Vesicles are used to transport materials within a cell between the RER, Golgi apparatus, and plasma membrane. The ribosomes attach to the RER synthesize proteins and release them into the RER. The proteins travel through the RER and are then released from the RER in the vesicles formed from the RER membrane. The vesicle travels through the cytoplasm and delivers the proteins to the Golgi apparatus, which then modifies the proteins. The proteins are then sent off inside a vesicle formed by the membrane of the Golgi apparatus and carried to the plasma membrane. The vesicle fuses with the plasma membrane, and then contents are released outside the cell, which we call the exocytosis, as you can see from the picture. So this is the RER where the proteins are made, then the made proteins are sent to the Golgi apparatus, and the Golgi apparatus modifies them, then send them inside the vesicle to other cells. So particles are in constant random motion in liquids and gases. If the particles are impermeable to the membrane, they can diffuse across. Particles will move from an area where they are in high concentration to an area of the lower concentration. Once the concentration of the particles on both sides of the membrane is equal, the particles will continue moving, and this state is called equilibrium. A concentration gradient can be defined as an increase or decrease in the density of the chemical substances in an area. Cells often maintain concentration gradients of ions across their membranes. In other words, it is the difference of the concentration, whether high or low, of a substance on either side of the plasma membrane. Diffusion. So you know that, guys, water is able to move in and out of the cells freely. Sometimes the number of the water molecules moving in and out is the same and there's no net movement. But at the other times, more molecules move in one direction. So the diffusion is actually a spontaneous tendency of substance to move down its concentration gradient from a more concentrated area to a less concentrated area. This movement of particles happens as a result of the random motion of particles and this existence of a concentration gradient. We can experience it with during the lesson. So what we're gonna do, we're going to actually take the two flasks, and one of them will be with a little water, and the other flask will be in a high concentrated of the water. So if we just add like a paper tissue between them, you will see if we color the waters, you will see that the water will go from the higher concentration to a lower concentration. Let's see a video how actually diffusion works.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
So, alright guys, this is actually the animation of how diffusion actually works, and there are actually questions here about simple diffusion as it's defined, and there are certain questions that you can easily answer. So now I'm gonna just like put it here. The diffusion of water across a selectively permeable membrane is called osmosis. Osmosis is due to the differences in the concentration of the substances dissolved in water. The movement occurs from an area of higher concentration to an area of lower concentration. Water can move by osmosis across the plasma membrane in both directions very easily because the plasma membrane is very permeable to water. Now let's see how the osmosis actually work.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
So, okay, this is the how actually osmosis work, and they're the questions that you are going to answer. So, now let's get to the other part of our lesson. So, particles move across membrane by simple diffusion or facilitated diffusion, osmosis as active transport. So, we're going to actually learn today the only simple diffusion as you see here. So, osmosis may occur when there is a partially permeable membrane, such as a cell membrane. When a cell is submerged in water, the water molecules pass through the cell membrane from an area of low solute concentration to one of the high solute concentration. Aquaporin is an integral protein that, as its name suggests, acts as a pore in the membrane that speeds up the movement of water molecules. Osmotic balance is a form of the dynamic equilibrium, also known as a water balance, where the obtained where there is many water molecules moving into an area as they are moving out. Effects of osmosis. Cells will react differently to different solutions. Plant cells and animal cells will also react differently to different solutions. There are three types of solutions that have an effect on cells: isotonic, hypotonic, and hypertonic solutions. What is an isotonic solution? A solution where there is a concentration of solid molecules outside the cell is equal to the concentration of solid molecules inside the cell. So the concentration of the outside and insides are the same, so we call it isotonic solution. Isotonic solutions are the ideal solution for the cells to be contained in. A cell will be at equilibrium in this solution. Whenever we get injection, we always get injection with an isotonic solution. For example, isotonic solution of natrium chloride, isotonic solution of different molecules, different injections. So remember, this isotonic solution is very important for the homeostasis. When it comes to a hypotonic solution, what is a hypotonic solution? A solution where the concentration of solids outside the cell is lower than the found inside the cell. So it means that the concentration inside of the cell is more than the outside. So cells placed in this solution will not be at equilibrium and it will experience a net movement of water inside the cell by osmosis. This will result in increased pressure inside the cells. Remember, guys, if the concentration inside is more, so it means that the cell will give out the concentration. So remember, the net movement of water inside the cell will experience a net movement of the water inside the cell by osmosis. So the solid concentration is greater inside the cells, the free water concentration is greater outside. So the free water concentration will get into the higher concentration of the cell. So free water flows into the cell where there is a higher solute concentration. When it comes to hypertonic solution, it's like a solution where the concentration of solids outside the cell is higher than the found inside the cell. So a cell placed in this solution will not be at equilibrium and it will experience a net movement of water out of the cell by osmosis. This cytoplasm of the cell will shrink due to the loss of water. The shrinking of the cytoplasm in plant cell is known as plasmolysis. This is the hypertonic solution. So because the external environment is in a higher concentration of the solute, the inside of the cell, so it's not like that. So inside of the cell will give out the water and so it will lose water. The free water flows out of the cell, which we call hypertonic solution. So isotonic solution is like that. Nothing happens. The outside and inside environment is the same with the water concentration. When it comes to hypotonic solution, it means that inside the cell there's a higher solute concentration, but outside there is more water, so the water will move into the cell and the cell will become bigger and it will swell. When it comes to hypertonic solution, it means that the outside, the external environment is full of the higher concentrated solute, so the water will the cell will lose water, so the water will flow out of the cell, and that's why the cells will shrink. So the importance of the osmotic control. So remember guys, also tissue and organs can be used by these isotonic, hypertonic, hypertonic solutions. In plant and animal cells it's very different. So if the cells shrink when there is like the water is going out to the higher concentration, it's gonna be flaccid. When is like isotonic solution for the plant is not good because it's gonna be flaccid and plants are gonna wilt. But the turgid is like when there is more water outside and it's getting inside, so then it's gonna be turgid. So the plants need more water, but in the animal and human cells it's very different. Isotonic solution is important for the homeostasis. Hypertonic is that when there is outer external environment is full of the higher solute concentration and the cells are going to lose water. And the hypertonic is that when there is a more concentration in the cell, so the water will flow to the cell and it will swell. So osmotic pressure. Animal cells cannot withstand a high amount of osmotic pressure and the plasma membrane will burst. Osmotic pressure inside the plant cell is known as a turgor pressure. Plant cells can stand up to the pressure due to the presence of the cell wall. Turgor pressure helps to keep a plant cell rigid. Now review part, you're going to answer this question. If you cannot answer one of them, go back to the presentation slides again, reread and try to answer this question later. Thanks for watching guys. Have a nice day. Bye bye.